Live now.
So I actually had one positive feedback so far.
There are many students in a discussion forum. So there is one faculty professor. She said that she's a follower of your topic. She has given very good comments. She is from physics department, professor.
Okay, we are early.
Hello.
And they'll come and join you.
Okay, so right now there's nobody there.
I don't think so.
We have to wait. I hope not. Water.
You shared the details, right?
Yeah.
Check Karthik. Where did I get my bag?
What are you looking for?
I'm looking for my bag.
Bag.
It's there. Not. No.
So
Karthik. Wait. Now we are going live with Harry's course. You are able to see that? Okay. Is there anybody online? Or? Can you see somebody online there? Okay, correct. That is fine. But any other students?
hi Karthik.
Anjita, there are 5 people already.
Oh, already. Okay. So what should we do?
We are unable to see them. Okay. But they are expecting more people. 5 people are already there.
Okay. So they can see
I
us.
couldn't see. Okay. We are already live, I think. Yeah, there are 7 watching.
Here in the side they are showing.
Yeah, there are 7 watching.
Here in the side they are showing.
Hi. So welcome to the live session. I think there were a few questions that were asked in the spreadsheet, and here is Anand. Anand, come here. This is Anand.
Hi.
He's been answering all your questions on the forum. So we have at least 3 or 4 questions that have come up, and so we'll go through them one by one. So the first question that we had was, the important question that we had was, what is the pattern of the exam? And so the exam pattern is, it's a multiple choice question. Karthik, can you hear us?
Yes.
If you can hear us, then please say yes.
Yeah.
Okay. So it is basically multiple choice questions, and so the rest of the exam questions are, rest of the formats are the same as other NPTEL. And so now to the scientific part. The first question that we had was, what is the difference between an antagonist and an inhibitor? Okay. So the here, just to recap, if you have a receptor on a cell surface, then it's going to interact with a ligand, and that's going to transmit a signal. Now, in this context, is where we look at what is an agonist and what is an antagonist. So, an antagonist is some molecule which will go and compete with the ligand and sort of prevent the signal from being transmitted. So, an inhibitor on the other side is fundamentally they are similar in nature. That means that the inhibitor is going to go and bind to the enzyme, and it's going to, depending on the nature of inhibition, we have spent quite a bit of time on trying to look at what are the various kinds of inhibitors. So there's competitive, non-competitive, non-competitive, so on. So these are, fundamental nature of inhibition is that they're going to go and bind to the enzyme and going to be, prevent the enzyme from binding to the substrate. So in some sense, there are similarities between the two concepts. But the important difference is that the antagonist will go and interact with the receptor and prevent the signal from being transmitted. And the inhibitor will go and bind to the enzyme. Can be an allosteric inhibitor or it can be an active site binding unit. But either way, it is going to prevent the enzyme from turning over the natural substrate. Okay, so I hope that answers your question. If you have any further questions, you can always ask on the chat portal. We will discuss further. Did I miss anything, Anand? No. Okay. So, what he has written is that all antagonists are inhibitors, but not all inhibitors are antagonists.
Yes.
So, because inhibition is something that is very common to enzymes. Okay. So, that is the first question. The second question was about structure-activity relationships of drugs, SAR of drugs. So, there was no question really, but this was asked for me to explain what SAR is. So SAR is nothing but structure-activity relationship. So what we try to understand in structure-activity relationship is, if I make a modification to the drug, how does it affect the activity? So if you think about the same example that we looked at previously, let's say we're designing a drug as an antagonist. Okay? And now the antagonist must go and bind to the receptor, and it must inhibit the natural ligand from coming and binding to it. So if we make some changes to the antagonist, how will it affect the activity? So now if we do a systematic analysis of this, that means that I increase the chain length, or I increase the hydrophobicity, or I increase the molar refractivity index. What we looked at all of these patterns, these parameters previously. So all of this can be systematically changed. If we do this kind of analysis, it is called as quantitative structure-activity relationship, where we try to assign a value or a coefficient to the parameter. Okay? So broadly speaking, structure-activity relationship is basically a large portion of medicinal chemistry is basically understanding structure-activity. I hope I have answered this question. Again, if you have any further doubts, we can discuss on the portal. The third question is about why is uracil present only in RNA and not in DNA? So I will let Anand answer this question.
Okay, to answer this question, one thing which you should be knowing is in case of RNA, it contains uracil, but in case of DNA, it is thymine. Making the thymine is energetically very costly, and therefore RNA prefers to use uracil because RNA is a very evolutionary old molecule when compared to DNA. And other cases, this uracil can get accumulated in case of DNA, and it can lead to mutations. And there is an enzyme called as uracil DNA glycosylase, which chops off this uracil. But this enzyme is absent in case of RNA, and therefore RNA prefers more to get uracil instead of thymine. So that is one major reason why RNA prefers uracil and DNA prefers thymine. I hope this answer is sufficient.
So just to put this in perspective, idea that we're still trying to understand the origins of life. So as we know it on this planet. So one of the ways in which life originates or proposed is that there was a complex soup in which there were all these biomolecules or these molecules which later became biomolecules were present. And one of the theories is that RNA was the first important molecule that came up. Then it was followed by DNA. Right? So here, one thing that is interesting about RNA is that RNA, we have again looked at in our course. And RNA is not just, it can also be a catalyst. It not only transmits information in terms of mRNA, but it also can do catalysis. So therefore, RNA it is suggested that RNA came before DNA. So one of the problems with RNA is the lack of stability, and therefore there are certain structural elements in the DNA which are going to help with improved stability. So that is what Anand was trying to explain to you with the concept of uracil.
So there is another question: what is the difference between 2D QSR and 3D QSR?
2D. Okay, so we will take the next question, which is synergy. Before we get, what is the difference between 2D and 3D?
So this can be explained in terms of topology. Like for example, in case of
is it 3 dimensions?
Yeah.
In terms of how the is it like a plot of 3 dimensions?
No, it is not. We take 3 representative characteristics to explain the required result for the
Correct. So which means that there will be 3 axes
Yes,
on the
3
thing.
axes,
So it's one layer of 3D QSR is one layer more complex than 2D. Right? It's going to be more
quantitative
in that respect. So we really haven't covered this in our class. So I feel that it is a little bit out of the scope of our course. We have only covered QSAR in our course.
Yes.
So therefore, I would recommend that you go back and look it up and learn a little bit more about it, because it's a fairly advanced topic which we are not taken up in this course. So the next question is, what is need some explanation on synergy? Okay. See, synergy is basically when we have 2 drugs, A and B, and if they're administered to a person, now the effect of A alone and effect of B alone is going to be some value. Right? But now what you would expect is that if I give A and B, the effect would be additive. That means the effect of A plus effect of B. But sometimes what happens is that the presence of A greatly increases the activity of B. And one of the ways to think about this is that, let's say there is an enzyme that breaks down molecule A. Now, if that enzyme is inhibited by B, then one way to think about it is that the activity of A is going to go up. But B itself might have its own activity in addition to the sort of protecting A, but B itself might have its own activity, and that also contributes. So people are very interested in finding drugs that synergize with one another, because that reduces the dose. So you can give a much lower dose of the drug and get a better activity. Is there anything else?
In addition to this, I can tell you one live example. For example, In case of bacterial infection, we prefer antibiotics such as penicillin. Like you can take amoxicillin. So this amoxicillin inhibits the cell wall biosynthesis, but it is required at a very higher dose, around 700 or 800 mg. So therefore, at the same time, bacteria also becomes resistant by producing beta-lactamases. So what the scientists thought is we will give a beta-lactamase inhibitor, such as clavulanic acid. So it reduces the dose, as Hari suggested, that we can give in a maximum dose up to 500 mg. And this will also overcome the resistance produced by the bacteria. So this is a very good example of synergistic effect.
Right. And in this particular case, the molecule that is given along with the antibiotic is actually doesn't have very strong activity.
Yes.
But there are other cases where you can give two drugs in combination, which has a better activity than the two drugs alone. Right? So there is a mathematical way of understanding this also. We again did do this in our class because it's a little advanced topic. There's something called as fractional inhibitory concentration that one can determine. Again, I think it's a very advanced topic, so we can probably discuss this offline. Or you can go back and look it up. There's something called as FIC, fractional inhibitory concentration. And there's a systematic way of studying whether a drug synergizes with another, whether it's an antagonist, or I'm sorry, whether it inhibits the other drug and so on. So those kinds of studies can be done. Any other questions? We don't see any other. We don't see any other questions on the portal. So I'm wondering. I hope we have addressed the points that have been asked. Okay. So Karthik, if there are no further questions, can we say goodbye, wrap up? Karthik is on.
Actually, we have read on the syllabus before exam.
Log P problems. Okay. Can you how to solve it? So log P, I mean, it's basically the partition coefficient. So you measure the amount of the drug in octanol versus the amount in water, and that the logarithm of that thing is going to give you the log P value. So what we use it for is to understand, in terms of if you're going to do quantitative structure-activity relationship, we plug it in the equation and find out whether the log P plays any role in the efficacy of the drug. So here in this particular syllabus that we're looking at, it's important for you to know what log P is and how it is interpreted. We're not really looking at quantitative structure-activity relationship at a slightly advanced level. So I wouldn't worry about trying to sort of solve any problems related to log P at this point. Okay, what you need to know is that it's determined in the following manner, and you can use it to find out whether a compound is permeable or not, whether it's an important factor in binding or not. All these questions can be answered around log P. And there are other concepts in log P, for example, like something that gets across the blood-brain barrier. So we have discussed this in the course previously. Like, if you want something to get into the brain, then you need to maintain the log P at a particular level. I think it's below 3. I think.
Below 3, yes.
And then if you don't want it to get into the brain, then you keep it above 3. So all these things are done so that you can direct the drug better to the organ of interest. I hope you have answered this question.
Is there any negative marking?
I don't think so. Did we put negative marking?
Question number?
No, I don't. There is no negative marking. No. To my knowledge, there isn't. But you will see the question paper in a couple of days. Any other questions? Okay, I hope you all enjoyed the course, and we can. I'm sorry we didn't do more live sessions. I was quite busy teaching my regular course this time. So thank you very much once again, and I look forward to interacting with you in some way, shape, or form in future. Bye.
Bye-bye.
Or Dr.